Hi everyone, and welcome to this brief tutorial on the HALO study. Today I will be showing you how to process the blood you collect from each patient and ship it back to us. Each site will be receiving bags like this for each blood draw. So inside each bag there will be 4 sterile transfer pipettes, 1 discard tube, which is the one with the colorless cap, 1 tube with citrate in it, this one has a blue cap, 1 tube with citrate and benzamidine, this is the one with the blue cap with a red dot on the label, 1 tube with heparin, which has the green cap, a tube with procoagulant for serum, this is the one with the red cap, and 8 cryogenic freezing tubes. Immediately after collection, the 2 blue tubes and the green tube need to go on ice. The red tube must be placed vertically upright for an hour to allow the blood to clot. Do not discard the discard tube right away. This will be used to balance the other tubes in the centrifuge. The tubes placed on ice must be processed within 2 hours of blood collection. After taking the tubes off the ice, invert them a few times to mix the samples. Place the 2 blue tubes opposite each other inside the centrifuge, and then place the discard tube opposite the green heparin tube. You will be centrifuging at 1700 times G for 10 minutes. Now, this particular centrifuge requires a speed of 5,000 RPM for these particular tubes, but if you have a different centrifuge, you should either figure out the speed required to get 1700 times G or send us the radius of your centrifuge so that we can figure this out for you. While centrifuging, separate your freezing tubes by color. The tubes should look like this, with the blood separated out into 3 layers. There will be a red blood cell layer on the bottom. On top of the red blood cell layer, there is a thin layer called the buffy coat, which may look white, and it consists mostly of white blood cells. On the very top, the largest layer is the plasma that you will be collecting and placing in the freezing tubes. When extracting the plasma from each tube, use only 1 transfer pipette per tube. Make sure that the plasma from the tube with the blue cap, without a dot, goes into the colorless freezing tubes. The plasma from the citrate benzamidine tube, which is the blue capped tube with the red dot, goes into the freezing tubes with the red dot on them. And the plasma from the heparin tube goes into the freezing tubes with the green dots on them. Take as much of the plasma from each tube as you can, but make sure not to extract any of the red blood cell layer or the buffy coat. After collecting the plasma from all 3 tubes, cap the 6 freezing tubes, and now we will be moving on to the serum collection. As I said before, this sample should be placed upright vertically for 60 minutes at room temperature to allow it to clot immediately after collection. After these 60 minutes, if you are not going to centrifuge right away, place the sample on ice. For the serum tube, unlike the plasma collection, you must centrifuge at 1200 times G. Again, place your discard tube into the centrifuge so that it can balance the serum tube. Carefully remove the serum tube from the centrifuge and stand it vertically upright. Again, we will be collecting the topmost layer without disturbing the layers below, and we'll be using the last remaining transfer pipette and aliquoting into the freezing tubes with the blue dots on them. Make sure all of the freezing tubes are screwed on tightly. Then place them back into the bag you received from us and store at minus 70 degrees Celsius before shipping them back to Hamilton. So at this point there should be 8 tubes in your bag. Store at minus 70 degrees Celsius until shipping, and once shipping, make sure to ship them on dry ice.